Welcome students. Today we are continuing from part 1. If you haven't seen part 1, check out the link in the description down below. Today's video, we will cover part 2, heredity and genetic technology. There will be multiple videos about EmSAT past papers. Make sure to subscribe to receive the next part of this past paper by turning on the notification bell to be notified whenever I upload a new video. Let's start solving these questions. The first question. The following pedigree represents the inheritance of an autosomal recessive syndrome in a family. If the individual 3 in generation 2, they mean this one, marries to a normal female who is heterozygous for the trait, what is the possibility to have an affected child? So we will not guess it in this way. Let's go and do it in a Punnett square to get the right answer. Let's answer it. As they mentioned, the male homozygous recessive, so I chose the letter B, B small, and the female, they assume that she is heterozygous, B capital B small. Let's draw the Punnett square. And now I'll distribute the alleles. Regardless, I will put the male up or the left, or the female up or the left, the same. So let's distribute the alleles normally. Here I put the female up and the male at the left side. The same rule, we have to look to the left side and the upper side for each square to get the genotype. So the 4 genotypes are, the first one, B capital B small means heterozygous. The second one, up and left, B small, homozygous, recessive. The third one, hetero. And the last one, also homozygous recessive. Once they asked us about the possibility to have affected kids, what we get here that half of the Punnett square affected. So the right answer for this question is 50%. Let's read this question. The diagram below show a part of central dogma of life in living organisms. Use this diagram to answer questions A and B. Actually, central dogma, it's the journey of changing DNA codes to protein, which includes 2 processes, transcription and translation. And if we just focus with this diagram, we'll find this is a translation. Actually, this is the ribosomes, and this is the messenger RNA. This is the transfer RNA that brings the amino acid to translate messenger RNA. Let's read question A. The process shown in the diagram is, choice A, is used to produce another copy of DNA? Sure not, because this is not DNA replication. B, copies the genetic information from DNA and carry it to the cytoplasm? No, that is transcription, which happens already inside the nucleus, but this is not. C, uses messenger RNA sequence to produce protein? Makes sense. Translation, but let's look at the choice D. Uses DNA and messenger RNA to produce ribosomes? Actually, even this statement is wrong. So the right answer here is the choice C, which is a translation. Now, question B. If the codon C, they meant this, labeled on the structure B is the last codon that is coded by an amino acid, what will be total number of amino acids in the polypeptide produced? Simple, easy. We will pick the answer from the same diagram by counting. Counting what? If we assume that C as a codon is the last one, that means this will be the last amino acid. So if we count how many amino acids we have, 1, already we have here 2, 3, 4, 5, 6, 7, and 8. So this polypeptide chain will contain 8 amino acids. The right answer here is the choice B. What about this question? Scientists claim that environment can play a role in determining the phenotype of an organism, and that's right. Not all traits should be inherited from the parents. Which of the following statements provides the best evidence for this claim? Choice A. A primrose develops white flowers when grown above 32 Celsius degree and red flowers when grown at 24 Celsius degree. Actually, the temperature is an environmental factor. But let's continue and see the other choices. B. The more genes involved, the more continuous is the variation in phenotypes. That's right, but where is the environmental factor? So this is not the right choice. C. Through breeding, red and white flowered 4 o'clocks produce pink flowered offspring at any temperature. Where's the environmental factor? 4 o'clock? No. It's not a season even. They did not mention it, and they ignored the temperature. D. A curly-haired Caucasian and a straight-haired Caucasian will have wavy-haired offspring regardless of the time of year. Even here, they did not mention the environmental factor. So we will choose the choice A because A includes the temperature, which is an environmental factor. So this is the right answer. Let's see this question. Uncontrolled cell division of cancer cells forms tumors. Which statements describes the difference between a cancer cell and a normal cell? Choice A. Cancer cells do not undergo cytokinesis. That's wrong. Any cell division should include cytokinesis, which is the division of cytoplasm. Choice B. Cancer cells do not have metaphase. How come? How we will divide the cell without metaphase stage? Choice C. Cancer cells replicate their DNA in interphase. Come on. Both cancer cells and normal cells will replicate the DNA in interphase, particularly in the S phase, I mean the synthesis phase. So this is not a difference. Choice D. Cancer cells have a shorter interphase. Yes, this is the point. Cancer cells spend less time in interphase and reproduce rapidly before the cells have had a chance to mature. Why? We have negative regulators of the cell cycle. Maybe it's less active or even non-functional with the cancer cells, as we see here in the figure, despite that we have the negative factor, but still the division of the cells go on. But for the normal cells with the negative growth factors, no division, and with the positive growth factor only, we have the cell division. So the right choice here is D. Now, the process below shows DNA replication. Which of the following letters represent the helicase enzyme? Many letters we have it here. And we know in replication, main enzymes like helicase, DNA polymerase, ligase, but we all know that the DNA helicase catalyzes the disruption of the hydrogen bonds that holds the two strands of double-stranded DNA together. And if we focused here, we will find that Q letter is representing this enzyme, and the other enzymes doing different functions. So helicase, who catalyzes the disruption of hydrogen bonds, is the Q. So the right choice here is D. This question is talking about what? Let's read it. Refer to the image below and identify the processes W and Z. Actually, from DNA to messenger RNA, this is transcription. And from messenger RNA to polypeptide, this is translation. This is process, if you remember, the central dogma. Let's read the choices. Choice A. W is replication, Z is translation. Z is right, W is wrong, because replication is not here. B. W is transcription, that's right. Z is replication. Again, replication not in this process. C, W is translation. I will not continue. Z is a translation, so I will cancel it. Let's go for D. W is transcription, and that's right. Z is a translation. That's good. So the right choice here is D. Now, this is about blood heredity. A man with type A blood and a woman with type B blood have a child. If this child has type O blood, which of the following statements must be true? Let me explain something. The heredity of blood, called codominance, what does that mean? Both A and B antigens are strong enough to express themselves. That's why, according to ABO system, we have A, B, and AB, and then we have O. O means no antigen available on the surface of red blood cell. Now, I'll explain it. I want to simplify it to show you how we received a child with O despite his or her parents having A and B. I mean antigens. Come with me, I will use the Punnett square. Let's explain the question. As they mentioned, that the mother, she has B blood, the father, A blood, and the child, O blood. Maybe you will ask, why I did not put the homozygous form, B capital, B capital, A capital, A capital? Because the child has two alleles without antigens, so one from dad and the other from mother. I will clarify it more and more using the Punnett square, distributing the alleles as usual, and using the rule of looking up and left for each square, we will find that yes, we have the chance to have a child with O blood without antigens. Depending on this explanation, let's go and choose the right choice. Let's read the choices. The father could be homozygous. A, never happened, because if we have it homozygous, we will not have any child with O blood group. Even choice B the same, I think. The mother has homozygous B, so cancel A and B. Let's read choice C. The maternal grandmother is homozygous. It could be, maybe, but how can I confirm it if they did not mention what is the genotype for the maternal grandfather? Let's move to choice D. The paternal grandmother could have type O blood. I will go with this choice. Yes, maybe C is right, maybe, but D 100% is right. So in this case, we are looking for the best answer. The right answer then is D. Let's read this question. Which of the following messenger RNA sequences will code for the following amino acid chain? Now, we have valine, serine, proline, tryptophan, and histidine. Polypeptide chain. Now we want to use the chart of amino acids. The rule of this is to decode the codon, we mean the messenger RNA, the choices here, we have to move from the center toward the periphery. Come with me, we'll explain it choice by choice. Here we will start. Let's move first to choice A. Let's start. GUG. Center the rule, so we'll look for G. Then moving to the periphery. This is U. And another G. This is G. So it should be valine. This is the first one. AGC. Again, center A. This is G. And then we'll move to C. So it should be serine. CCA. This is C. Another C. And moving to A. Proline. UGG. This is U. And this is G. Another G. Tryptophan. CAG. Go back again to C here. And this is A. And this is G. So it should be glutamine. This is for the first choice. Moving to the second choice. If you want, pause the video and try it by yourself. Then run the video again and compare your answer with my answer. We'll come back. Let's see, compare your answers with my answer. Valine, serine, proline, tryptophan, and the glutamine. Let's move to choice B. Again, pause the video, do your work, and compare your answer. Again, we'll come back. Let's see. Valine, serine, proline, stop codon, and histidine. I want to give a comment here. Once we received stop codon, this is wrong. What do you mean by stop codon? Stop codon means stop doing the translation. So without thinking, we have to exclude this choice. Let's move to choice D. Again, pause and do your work. Good, you came back. Valine, serine, proline, tryptophan, and histidine. And if we compared our work with the choices that they gave, we will find that the right answer should be D. Let's go back. So here, after doing all that work, the right answer is D. Please be more focused while you are doing this. What about this question? Let's read it. Albinism is an autosomal recessive disorder that affects melanin pigment production in skin, hair, and eyes. If both parents are carrier for albinism, what are the chances for their child to be albino? That means having albinism. Before answering this, let's explain certain terms. When we are talking about recessive, that means two small letters. We were represented by two small letters. And when we are talking about the parents are carrier, that means they are heterozygous. What does that mean? Capital letter with a small letter. So to make sure that 100% our answer is right, we need to use the Punnett square. Let's do it on Punnett square. Let's start. So, as they mentioned, the parents are carrier, so I chose the letter A to differentiate between uppercase and lowercase. Let's draw the Punnett square and distribute the alleles on the upper side and on the left side. As usual, we will look up and left for each square, up and left. So the first one, homozygous dominant. The second one, heterozygous, means carrier. The third one, also carrier. And the last one, recessive. So the percentage of having albinism is 25%. Let's go back and answer the question. So the answer for this question is D, 25%. This question has a graph. The given graph shows the change in DNA content during various phases from 1 till 4 of the cell cycle. Which of the following explains the changes to the DNA content at phase 2? So we are looking for this phase. Let's explain it using this figure. Now, this is the cell cycle, and we have 4 phases. Yes, we have here G0, that means the cell in rest position, but is still doing its function. Now, gap 1, I mean the G1, most of the time here, as you see, spends in the cell growth, replication of certain organelles. Now, moving to S phase, which is synthesis. This is replication of DNA. I think they mean this phase. Now, moving to G2, gap 2, finalizing the growth, finalizing replication of certain organelles, then it will be ready to go for M phase, which is the cell division. So, once they are asking the changes in DNA, the right answer should be D, replication of genetic material. It seems an easy question. Let's read it. A student is observing cells undergoing mitosis using the microscope. He notices chromosomes of some cells lining in the cell equator, like this. Which phase of mitosis is this student observing? Let's see. Actually, we learned about it a lot, that we have 4 phases for mitosis division: prophase, metaphase, anaphase, telophase. And we all know that when the chromosomes align at the middle, this is metaphase. It's very simple, I told you. So the right choice here is B. Here, another question about amino acid charts. Which amino acid sequence will be assembled from the messenger RNA associated with this strand below? And they gave us the DNA template strand here. Not directly, they give us the messenger RNA. Focus, please. So it's run from 5' to 3'. I'll explain it in details. And keep in your mind, we have 2 types of charts. The previous question, we solved it. It was using the circle one, and here we will use the table. All easy. Come with me. Let's answer the question now. This is the sequence of DNA that we want to do the transcription for it and to have the messenger RNA. But keep in your mind that on messenger RNA and on any RNA, we don't have thymine. And instead of thymine, we must have uracil. So when we want to do the complementary, according to Chargaff's rule that said guanine with cytosine or cytosine with a guanine, adenine with thymine, thymine with adenine, and instead of thymine, we have to replace it with uracil. Pause the video and try it by yourself. Then I will give you my answer. We'll come back. Let's see the answer. Exactly, as I mentioned, guanine with cytosine, cytosine with a guanine, adenine, and instead of thymine, we have to replace it with uracil. Thymine, it's okay, we have adenine here. Now, we will leave the DNA sequence and we will use the messenger RNA. We have 4 codons, each codon made up of 3 nucleotides. Now, we want to do the work. Let's do the work. This is another type of amino acid charts using tables. No problem, it's easy. Let's start with the first codon, CGC. The rule is looking for the left side first, then the upper side, then the right one. Let's start. We have C, so we are looking for this row. We will keep our eyes on this row. G, upper side. So this is G, moving all the way down here. So we were here and we will move here. So our eyes, we want to keep it on this square. And the last one is C also. Okay, that means the amino acid is arginine. Let's do the second one. Again, UGU, left, up, and right. Left, up, and right. The left, we will go for uracil, I mean. So we'll keep our eye on this row. Then up, G. So our work in this is square. Then right, this is another U. So it's UGU, which means cysteine. Pause the video and do the work by yourself for AUG and UGA. Welcome back. Let's continue the work. Now we have AUG, the same rule, left, up, and right. This is A. We will keep our eyes on this row. Then U, uracil, so we'll go all the way here. That means our work in this square. Then G, all the way from here, so we'll find it, methionine. Moving to the last codon. Again and again, left, up, and right. Uracil, guanine, and adenine. What does that mean? UGA, but it's a stop codon. It's a stop codon. What does that mean? The translation stopped. We will not add any amino acids. Keep this in your mind. Now we have our sequence. Let's go to the question answers and choose the right choice. Now to answer the question, we will compare our answer with the choices. So the right choice here is B, arginine, cysteine, and methionine. Yes, they did not write the stop codon, but this is the right answer. Let's look at this question. Which of the following is or are true for the process in the attached figure? It's easy. If we learn very well the process of transcription, we'll understand this is the pre-messenger RNA. Why I'm telling pre? Because still we have exons and introns all together. We know that we have parts of DNA non-coding. And this non-coding we should get rid from in order to have the right protein after translation. And here the parts of introns representing this part. So we need to get rid from introns. From its name, introns, they have to stay inside, and exons, they have to exit the nucleus. So by what? By splicing it using certain enzymes. We call this process also alternative splicing. So if we start reading the choices, we'll find the first one, it takes place in the nucleus. That's right. Exons are removed by the end of this process? Sure not, because we need exons. They will represent the structural genes. 3, it produces an messenger RNA that is ready for translation? Yes, that's right. So if you want to choose the right choice, it will be the choice D. This concludes the second part of our EmSAT paper. We have covered heredity and genetic technology. Next video, we will answer more questions on evolution and diversity of life, part 3. Make sure you like and subscribe and turn on the notification bell to receive more videos from Mrs.Biology. Thanks for watching.